Hello and welcome to the concluding keynote address on neurotechnology, making a bridge between basic neuroscience and clinical applications. Neurotechnology has seen a very dynamic period in recent years in fields such as neuroprosthetics or brain-computer interfaces, helping the modulation of brain activity. And it has made a meaningful impact also in neurotherapeutics. To address this theme, we could not have been more fortunate than to have with us Professor Pierre Magistretti. Pierre is a professor at EPFL and University of Lausanne, and also at the King Abdullah University of Science and Technology. He's a distinguished neuroscientist, but he's also active in the field of neurotechnology as senior advisor to Clinatech. In the past, he was president of FENS, he was president of IBRO, and served in many other important capacities in the field in many organizations. Now, when I ask a friend what I should, in addition, say about Pierre, he told me that Pierre is not only an outstanding scientist, but he's also someone who will go the extra mile, not just to lift the topics that he's interested in himself, but the entire field of brain research. So with that in mind, I would like to welcome very warmly Pierre Magistretti, and over to you, Pierre, for our concluding keynote address. Thank you.
Thank you very much, Paweł, for this very nice introduction. Thanks to Katrin and also as the leader of scientific leader of the Human Brain Project for inviting me and give this keynote lecture on how neurotechnology is really an emerging and even more than emerging, established now in some cases tool to bridge basic neuroscience with therapeutics. And in fact, I would argue that possibly the most striking advances in CNS nervous system therapies over the last decade, I would say, emerged from neurotechnologies. And we hope also that small molecules and pharmacology and biologicals will have an impact, hopefully. But it's currently really the advances have been very much from the neurotechnology side. So we have, let's have a look at the current situation. We have a population that is aging. We know there is an important burden of neurological and mental health, and so we really need innovation. The challenges are many, and it's really a worldwide problem. And again, the aging population with the risks that it adds for neurological disorders, in particularly neurodegenerative disorders, cannot be overlooked. Now, what are the challenges for brain diseases? Well, there is really a lack of efficient treatments for many diseases. Just take a big one, the elephant in the room, Alzheimer's disease. There is also a rising cost in drug development. So it's not an easy journey going from maybe a very promising observation in the lab where mechanisms of disease possibly have been identified, and then eventually having a drug on the market. Another reality is that big pharma essentially is divesting from research in neuroscience. It has invested billions of dollars over the last 2 or 3 decades, but results have been relatively meager, not zero, but let's say meager. And there are other topics that are really much more productive in terms of treatment, cardiovascular, metabolic, cancer, of course. So, and one can understand the choice. But the big challenge also is the economic burden in Europe. There are many studies, actually sponsored in some cases by the European Brain Forum and other European organizations. But there is also the human burden, of course, which is incalculable. And despite, in fact, in contrast to this kind of, difficult situation, the rise in knowledge in neuroscience is exponential. I mean, really, we know more and more the molecular, cellular, now even circuit level about how the brain signals, the brain works, processes information. So there is really a gap here. So how can we try to bridge this gap between increasing knowledge in neuroscience and the actual rather meager, collectively reached therapeutic approaches? Well, um, as I said, very little novelty in molecular entities. You can see great increase in cancer or cardiology, but very flat developments and new molecules in terms of brain diseases. This vesture of many companies, that's another reality. So, but there are in neuroscience aspects that really can be tackled mostly by these neurotechnologies. First of all, the natural electrical engineering interfaces. The big progress has been made. I will give you some example. Simulation also. Virtual reality can really be of help. Of course, the imaging, also great development in imaging, and also still not really reaching the clinics, but promising directions in optogenetics, and then other approaches that are less in the direction of neurotechnology. So let's look at the successes of neurotechnology, as I mentioned at the beginning. Well, maybe the main one is what Alim Benabid brought to us already at the end of the 80s and developed even much more in the 90s. And now, with more than 200,000 patients having benefited from what is now known the deep brain stimulation, mostly for Parkinson's disease, but later different movement disorders, in particular dystonia, and maybe also some psychiatric diseases, obsessive compulsive disorders, depression. The initial progress has been a bit uneven, but as you may know, recently there has been really at least an interesting study from UCSF showing effects on refinements of deep brain stimulation in depression. Another example, paraplegia. My colleague Grégoire Courtine and Jocelyn Blog at EPFL and the University Hospital of Lausanne, they have really provided modern proof of concept now that brain-spinal cord interfaces connecting stimulating the distal portion of below the lesion in the spinal cord. They have shown that really quite significant advances, with even some contribution from higher brain centers in allowing more or less a physiological walking. Of course, there is still work to be done, but it's really possibly the major progress that has happened over the last 2 or 3 decades with these neurotechnology approaches. And here the brain, computer, brain or BSI, also spinal cord interface, it's really a key. Of course, this has gone also with some material development and approaches to develop, for example, electrodes that can be very flexible, can be put in different areas of the nervous system, micro, of course, electrodes. This is again work at the Center for Neuroprosthetic at EPFL with Stéphanie Lacour. I'm going to mention sites which I know well, having spent as the head of the Brain Mind Institute many years at BMI, also at the University of Lausanne. again, as I mentioned, these soft electrodes can be interfaced in key points to control neuronal activity in different manners with different routines, depending on what one wants to achieve. So a lot of specificity. Um, Another development, a proof of concept for the time being, but very striking. Now we're talking about tetraplegia, not paraplegia, or even with some fibers left. This is total tetraplegia. And at Clinatech, again under the direction of Alim Benabid, a proof of concept with an exoskeleton controlled by epidural wireless brain-machine interface. This notion of epidural is very important. We're not talking about implanting electrodes within the brain parenchyma, so it's much less invasive. It can last for much longer, and so this is really a major breakthrough. Now, how that will be adapted to really improve from proof of concept to daily life, this is works in progress at Clinatech, which is really an amazing place, as I will mention in a moment, with a operating room and patient rooms next to a major technology center and with biology in between. Another advance that you probably already heard, and it's really thanks to the work done in particular in Katrina Moon's lab and with her collaborations, a really high definition 3D atlas about human brain cytoarchitecture, which can be extremely useful for neurosurgical operation. And this really is really the most advanced neurohistology, neuroanatomy device that can help in clinical work. Also, another development which is less known, which is emerging, but I think has a lot of potential, is photobiomodulation. And again, this is implemented in Clinatech by John Mitrofanis, who joined recently Clinatech from University of Sydney. And the idea is that there are many evidence indicating that light at specific wavelength interfere positively with mitochondrial function in particular, stimulating, for example, ATP production, energy production, and displaying clearly intrinsic mechanisms or enhancing intrinsic mechanisms that promote cell survival. So this is, I think, it's a very promising new area for neuroprotection and neurodegenerative diseases. And in fact, again, in the spirit of Clinatech, or in the two patients have been implanted with light probes in the lateral ventricles, and these are early stage Parkinsonian patients with the idea to actually slow down degeneration. Of course, these studies will take much longer than DBS, which is immediate in its effect, but it's definitely something that holds promise. And since we're talking about metabolism, let me now talk about my work, which has been over the last 3 decades concerned with trying to understand the mechanisms for coupling neuronal activity with metabolic responses in the brain. As you know, there is a tight coupling. By the way, this is the basis for all the techniques for functional brain imaging, PET, fMRI. They're based on the fact that there is a metabolic or vascular response in response to the electrochemical events that occur at the synapse. And over the years, in fact, my lab was the first to demonstrate that the key players in this coupling are non-neuronal cells, astrocytes, which overall, the glial cells make up half of the brain cells, and astrocytes are the major component. And so what we have shown is that that astrocyte can sense synaptic activity and translate it into a metabolic response, and I will tell you a few words about it. Just for those who know less about astrocytes, people know a lot about neurons, but astrocytes are very interesting cells. They are positioned between the source of energy, the capillaries, and just keep in mind that we have 600 kilometers of capillaries in our brain, and synapses. And not only they're at the right position, they also possess molecules that can detect synaptic activity, such as receptors and reuptake sites for neurotransmitters, in particular glutamate, the main neurotransmitter in the brain, and the source of energy, the capillaries, through this end feet, which also express proteins transporters for glucose and amino acid and other substrates. So what we described about 20 years ago is what is now known as the astrocyte neuron lactate shuttle. What we showed is that, in fact, astrocytes sense when neurons release glutamate, and again, glutamate is released at 80% of the synapses in the brain. And this uptake of glutamate, I will not go into many details, but essentially some of the glutamate is recycled back to neurons under the form of glutamine, and so to reestablish the neurotransmitter pool. But there is also an entry of sodium as the driving force for glutamate to be taken up. So essentially, these two processes of recycling glutamate and reestablishing the sodium gratins costs energy. And so astrocytes take up glucose from the capillaries, the endothelial cells, and process it through what is known as aerobic glycolysis, meaning the transformation of glucose to lactate in the presence of oxygen. It's known as the Warburg effect. And lactate, we and others have shown, is a very appropriate energy substrate for neurons, in fact, even preferred. Doesn't mean that there is not glucose going into the neurons. There is about 20% of the glucose entering neurons for different functions, which I will not elaborate. But during activation, this astrocyte neuron lactate shuttle is the main mechanism to provide neurons with energy. And as you will see in a moment, not only energy, but also some signals for neuroprotection and plasticity. So let me just show you an example where we have used, let's call it neurotechnology in the broadest sense, but in computational and modeling approaches with the team at EPFL from the Blue Brain and project. And what we have modeled is essentially the process by which another neurotransmitter, noradrenaline, acts on astrocytes to mobilize energy in another way. Astrocytes contain glycogen, the storage form of glucose, and noradrenaline is able to mobilize this glycogen and lead to lactate production. So we modeled this interaction with about over 40 differential equations for each step, and essentially validated many experimental observations. And so we have now a model on which to work to better understand this coupling. So again, this computational approach has been very useful. Another work that we have been done is another level of resolution from the atlas. It's just looking at single cells and reconstruct them digitally completely. This again is work by mostly Corrado Cali in my lab, Jay Cogan at EPFL Blue Brain project, and essentially reconstructed what we call the neuron glia vasculature unit, single neuron, single astrocytes or many astrocytes, and the capillaries. And the process is really starting with an EM block and then serially sectioning it and then reconstructing with appropriate tools. Eventually also, and I'll show you some examples, 3D quantitative quantification and reconstruction, and eventually digitize and introduce these digitized cells into a digitized model, in particular of a cortical column. So not only neurons, as was done before, but we have now introduced glial cells and vasculature. Now, here is an example of a human neuron reconstructed this way. This is work by Corrado Cali, who is now an assistant professor at the University of Torino. And he has made this kind of funny video where you see the modern and the old anatomy. This is the dissection room at the Institute of Neuroscience, which was the Institute of Anatomy in Torino. And he shows this human neuron reconstructed, the 3-dimensional, with a focus here on mitochondria and organelles, and then you can play with it and look inside at the intracellular organization. So it's well, this is really a bit for a demonstration, but it's actually very useful work. Now let's go back for a moment to this astrocyte neuron metabolic coupling. And a few years ago, in collaboration with Christine Alberini at NYU, we explored the possibility of lactate not being only an energy substrate for neurons, but also a signal. And there were many reasons why we did these experiments. And what we showed essentially is that if we block the mobilization of glycogen by noradrenaline and to produce lactate, or we block the exchange, the transfer of lactate from astrocytes to neurons, we block long-term memory formation. And again, we have expanded very much this work. But that was one of the first, if not the first demonstration that a clear role of an astrocytic molecule that was long considered, by the way, a metabolic end product is fundamental for one of the highest functions of our brain, which is memory. But we pushed, again, I just show you, rather than elaborating more on the physiology and behavior, we just show you some data, again, obtained with advanced neurotechnologies. And that's the reconstruction of a whole section, a block of brain, and then doing what we call segmentation. So on each section, you identify the profiles to which cell type they belong: neurons, glial cells, vasculature, or axons, dendrites. And so eventually, and that was really a major achievement done at KAUST, that was to transform this data into a 3D full immersion virtual reality. So this, the first phase is that, again, you can see here all the various elements, and in green, look at the astrocytes, how pervasive they are in this block. And then you can visualize in purple the glycogen granules. But here you have Corrado, again, actually taking a walk inside this block. He's in the capillary here, and these are real data. You can see down here the section, the EM section, and this is a 3D reconstruction and immersion, full immersion virtual reality. So now he's behind the capillary where he was walking in for a while, and now he's going to get into an astrocytes. And he sees many glycogen granules. He can see in transparency a dendritic spine, and so you can really achieve not only something nice and I would say spectacular, but really some quantitative data about where the glycogen granules are clustered, because that was our question. Since it seems that glycogen is so important for memory, where is it localized? And it turns out that it is actually localized in the astrocytes near the synapses. And we extended this work and showed that, following learning, and again, this was known in part, that during learning there is an increase in the number and size of dendritic spines. But what was certainly not known is that there is also an increase in the size and number of glycogen granules in proximity of the synapses where the dendrites have been enlarged because of learning. And if one blocks glycosylation with this drug, does not lead to an increase in the number and size of synapses, nor in the, and that's more expected, in the glycogen granules. In fact, there is a decrease. And we can rescue, in part, with lactate, in particular, the number of dendritic spines. So clearly, lactate is important for synaptic plasticity and learning and memory. So let me now move to the conclusion here and say, what kind of environment can we promote to even go back to the first question? How can we treat patients? At the end of the day, personally, I feel very concerned. I am a medical doctor. Then I got involved in neuroscience, starting with a PhD, and then went on in neuroscience. But still, today I would dream that whatever I have found and my colleagues find can be useful to treat patients. And so one has to create an ecosystem for what we call neurotranslation. We have clearly necessity to have access to clinical space. This is very important. This is what has been achieved at Clinatech for sure, and at EPFL with the hospital in Lausanne. We need accelerators. In Geneva, there is the VIS Institute, at Harvard also. These are very important organizations to help accelerate and eventually lead to startup incubators, and then attracting large corporations, and eventually mobilize the interest of VCs. It's an alternative road to the classical pharma road, and I think it's particularly conducive and useful for neurotechnology. Now, in the next 2-3 slides, let me just give you very quick examples of 3 places I know well: EPFL, KAUST, and Clinatech in Grenoble. And EPFL, again, you have proximity between the academic and the innovation park. The hospital is not next, but there are units within the hospital where EPFL scientists work in conjunction with clinicians. KAUST, it's on the shores of the Red Sea, of an international university with really an amazing quality of scientists that have joined the university since over the last 10 years. It's only 10 years old. There is a campus, of course, the academic campus, the community, but there is a large research and technology park where startups and other companies, larger companies, are hosted. And then Clinatech is really a rather unique arrangement. It's next to CEA, Commissariat à l'Énergie Atomique, so a huge powerhouse in terms of technology. But then there is a section for basic research and technology, and labs for material sciences lab. And the real unique feature of Clinatech is that there is a clinical sector, an operating room, and rooms for patients. So this is really amazing to have basic research, clinical application, and huge technology competence next door. So to conclude, what are the main points that we should keep for reflection? Brain aging and mental health are really, and neurological diseases in general, really represent major human and economic challenges. There is a lot of progress in terms of understanding brain function and considerable knowledge acquired, but not much progress for novel therapies. Pharma has disinvested, and so there must be alternative players in this progress. Of course, government or organizations like the European Commission and other European organizations, they support already, but they ideally should even support more research such that academia can continue this research, again, connecting clinicians with engineers and scientists. So in that case, it's important also to develop new private-public partnerships models, how this can work. This is very important between industry, startups, and academia, and overall create a novel environment for translational research in neuroscience. And the Human Brain Project, eBrains, can certainly play an important role in this. Thank you for your attention.
Pierre, thank you very much for this both overview of the main most promising projects in the field of neurotechnology, also for presenting your impressive work in brain energy metabolism, and finally for taking us inside the brain, showing us this fascinating VR technology, which we should probably have here at the next HBP summit, and talking how you see the ecosystem for neurotechnology developing. We really have just about a minute left before we conclude the summit. And if I may, I would like to ask you about what you said with regards to the ecosystem for neurotechnology, because what you showed us from access to clinical space through accelerators all the way to VCs, I mean, this is a complex construction, and there are only a handful of places that are in the position to do this. You yourself believe that we should link different efforts in Europe. What would be the value added, and how do you think this could work for the benefit of advancing neurotechnology? If you could be very brief,
Well, I
because
think,
we literally have a
yeah,
minute.
I'll be very brief. I think one way is really, I think e-brains can play a role by, for example, connecting neurotechnology platforms across Europe. And you and I and others have been discussions on this. I think we definitely should promote this. And then on Tuesday, when we had the summit, the first session in person, where I was able to be in Brussels, I came up with this kind of maybe iconoclastic idea. I don't know, but when you see the success of EMBL in cancer, when you see the success of CERN in particle physics, why not create a couple of places in Europe where you would have clinicians, engineers, and neuroscientists together?
Brilliant, Pierre. Thank you very much. This is wonderful food for thought that you are leaving us with. So thank you for your speech, and see you at the next summit. Now, in a few seconds, we will start a concluding session of this year's annual summit of the Human Brain Project. So please stay with us. Yes.